Hello everybody. Welcome to the session of Edunguru. This is Dr. Pankaj Kumar, your biology faculty, and in this sessions we will be discussing about some of the major questions that is very important from NET perspectives. I think by this time all of you are well prepared for your competitive exams. You have gone through the chapters, you have learned the concepts, right? Now it's time just to check that how prepared you are. Okay, so what we will do, we will just go through some of the major questions. The questions will reflect in your screen, right? These questions are labeled as per the easy, moderate and tough. That will be shown by the different color scheme as you can see in your the screen. And as the question is reflecting on the screen, all of you are supposed to answer those questions or practice those questions along with me, right? So this session not only going to give you the much needed practice, but also it will help you in checking yourself that where you are as far as NET examination is concerned. Okay, right. Let's talk about the very first question, and the very first question is from the chapter of reproduction in plants. Let me tell you that the chapter of reproduction in plants is a very important topic from NET perspective, on account of the fact that out of 90, 5 to 6 questions are asked from this chapter alone, right? So you can understand that how important or how significant this chapter is from NET perspective. Okay, right. Here is your first question. The first question is talking about that you need to figure it out, the incorrect statement in the given 4 options. The options are: option A, monosporic embryo sac develops from single megaspore mother cell. Option B, bisporic embryo sac develops from 2 megaspore cell. Option C, 8 nucleate monosporic embryo sac develops after 5 nuclear division in megaspore mother cell. Option D, 8 nucleate bisporic embryo sac develops after 4 nuclear division in megaspore mother cell. Now, in order to get the answer, we need to analyze all these 4 statements. You know, the types of embryo sac in the very chapter of reproduction in plants is very important. So the first and foremost thing is that what are the basis on which the types of embryo sacs is actually recognized. You see, in a megaspore mother cell, that is a diploid cell, right? The reductional division is going to happen. So normally what happens? A reductional division happens, the 4 cell is going to be formed, right? And out of all those 4 cells, the 3 will degenerate and 1 will eventually develop into, and that will give to the functional embryo sac, right? But depending upon the megaspore nuclei which is participating in the formation of the complete embryo sac, the different types of embryo sacs has been recognized. Okay, so if only one nuclei participate, we call it the monosporic embryo sac, right? But suppose if the 2 megaspore nuclei is participating in the process, what we will say? It is a bisporic. Okay. And if there is going to have a 4, then we have to say tetrasporic. But mind it, the catch in this question is that it is talking about cell. But as a matter of fact, what happens? That it is not the cell, rather it is the nuclei that participate, right? So have a look at the option number B. In the option number B, what says? That bisporic embryo sac develops from 2 megaspore cell, which is incorrect. Why it is incorrect? Because it is not the cell that participate, rather it is the nuclei that participate. Okay, so what is the key learning over here? The key learning, that is a very minor thing, but perhaps the most important one, that in a development of the embryo sac, eventually it is the nuclei that participate, not the cell. Because what happens? First of all, nuclei participate, and later on the cell wall formation happens. Okay, so that's why the correct answer for question number 1 is option B. Okay, right. Let's come to the question number 2. Tapetum cell divides by option A, amitosis, option B, mitosis, option C, endomitosis, option D, meiosis. Perhaps the easier question. But let's try to understand that first of all, what is tapetum? What is its role? The moment we understand the role, then the types of the nuclear division that is involved, it is very much evident. Now what is tapetum? You see, anther wall is enclosed by, or we can say it consists of 4 layers, right? What is the outer layer? The outer layer what we call epidermis. This is followed by endothecium. Then again it is followed by middle layer, right? And the innermost layer what we call it is a tapetum. Okay. Now just to understand that the entire covering of the microspore or the developing microspores, we can say, the nutrition need to be provided by one or other layer, and that particular role has been assigned to the tapetum. Why? Because you see, tapetum is the innermost layer, and just below the tapetum what we see, there is a microspore cell over there. Okay. So the very function of the tapetum is to provide nutrition to the developing microspore, right? Now, since they have to provide nutrition, so naturally the choice of cell division is like that whatever the genetic material that is present within that particular cell has a capacity to form more and more food or more and more protein, right? So that is why the division that happens in case of tapetum cell is the endomitosis. Now what happens in endomitosis? You know, in case of endomitosis, the chromosome number gets doubled. Okay, so there is no division of the cell, or we can say there is no cytokinesis. Karyokinesis happens, but there is no cytokinesis. So what it means? It means that the genetic material is going to be doubled. And if the genetic material is going to be double, it means there is a more genetic material, more protein synthesis. And if there is a more protein synthesis, the nutritional requirement of developing microspore mother cell or microspore is going to be fulfilled, right? So that's why the correct answer for question number 2 is C, that is endomitotic cell division. Okay. Let's come to question number 3. To form mature embryo sac, the functional megaspore undergoes option A, 2 mitotic division, option B, 2 meiotic division, option C, 3 mitotic division, option D, 3 meiotic division. Now once again, you see, this is the most common and popular question which is frequently asked in competitive examinations. Now let's try to understand the entire development or the very process of sporogenesis. Now this question is talking about the formation of functional megaspore. Okay. So the formation of embryo sac from functional megaspore, we need to identify the starting point and the ending point, right? So the question is talking about functional megaspore, mind it. So megaspore, if I say, megaspore is always haploid, right? Now if megaspore is haploid, then what will happen? First, there is a 3 consecutive mitotic nuclear division. So when there is a 1 nuclear division, then what will happen? That 2 nuclei is going to be formed. Again there is a second mitotic division. Then we have a formation of this 4. Again we have a third mitotic division. So at the end of third mitotic division, what we see? That 8 nuclei is going to be formed, right? So what we are seeing, that here the formation of the functional megaspore, because we know very well that here after this the cell wall is going to be formed, right? And this structure will be formed. Okay, and this is what we call embryo sac, and we know very well, as far as embryo sac is concerned, we say it is a 8 nucleate but 7 celled structure. Okay, so as far as the division of or formation of embryo sac is concerned, the required division is the 3 mitotic division, right? So the correct answer for question number 3 is option C. But here I would like to draw your attention one very important facts regarding solving such questions. The key of solving this question is just one thing. You need to concentrate on the starting point. from where actually we are starting. For instance, here we are starting from the megaspore. Okay, but suppose it would have been megaspore mother cell, then the answer would have been 1 meiotic and 3 mitotic division, right? So in likewise, you need to see that what is the starting point and what is the ending point. Accordingly, you need to answer such questions. Okay. So this is all about question number 3. Let's talk about question number 4. Now question number 4 is talking about chalezogamy. Now question says chalezogamy is observed in option A, cucurbita, option B, casuarina, option C, pisum, option D, lycopersicum. Now once again, let's try to understand that what are this chalezogamy. The very interesting thing is that, the pollen tube always enters through the micropyle only, but the manner in which the pollen tube enters, the 3 types has been recognized. Okay, what are they? They are chalezogamy, mesogamy and porogamy, right? If I say chalezogamy, that means the entry is going to occur through the chaleza. If I say mesogamy, that means entry is going to occur through the integuments. And if I say porogamy, what it means? That entry is going to occur through the micropyle. Okay, but once again, I would like to make you understand, eventually the pollen tube enters through micropyle only. I can show you in the diagram. So suppose this is a pollen tube. Okay, so if the pollen tube is going to enter like this, right? So pollen tube is entering like this, you can see there, and eventually enter this. So this is actually called as a mesogamy. Okay, if the pollen tube is entering from chaleza, again it is along with the central structure, it will enter like this. Okay, so this is what we call chalezogamy, right? And as far as porogamy is concerned, then it will directly enter over there. Okay, so this is what we call porogamy. So the final entry always occur through micropyle only, but the manner in which the initial entry is going to happen, the 3 types has been recognized. Okay, so as far as chalezogamy is concerned, the typical example for chalezogamy is a casuarina. So in casuarina is a xerophytic plants. In case of casuarina, what we observe, that chalezogamy, right? And if you take an example of mesogamy, then mesogamy is going to occur in case of cucurbits. Okay, and as far as the porogamy is concerned, so that is a very common one. So most of the common types in which the pollen tube is going to enter, that is through the porogamy only. So the correct answer for question number 4 is option B. Let's talk about question number 5, which is the first requirement for pollen germination. Option A, presence of indole acetic acid and gibberellin. Option B, high relative humidity. Option C, optimum temperature. Option D, presence of calcium, magnesium and potassium. Now once again, let's try to understand the very sequence of events which leads to germination of the pollen grains on the stigma. Now the first and foremost thing which initiate the very germination of the pollen grains is the relative humidity. It is the relative humidity that determines the very penetration of the intine in the form of the pollen tube that comes out of the colpus. Okay, so it is the relative humidity, right? Once it penetrates and once it's entered into the style, okay, then the growth of the pollen tube is actually marked by the requirement of indole acetic acid. And again, there is a 2 more requirement. One is the calcium and second is the sugar. So these are the requirement that is required for the growth of the pollen tube. But as far as the germination is concerned, mind it, it is the relative humidity that determine the germination of the pollen grains on the stigma. Okay, so the correct answer for question number 6 is option B, that is high relative humidity. Let's talk about question number 6. A young pollen grain is best defined as option A, spore mother cell, option B, male gametophyte, option C, partially developed male gametophyte, option D, mature male gametophyte. Now again, this is one of the very popular questions which is frequently figured in one or other competitive examinations. We need to understand that what is gametophyte, be it a male or be it a female. Okay. So similarly here the question is seeking information that what is male gametophyte, or define pollen grains in terms of whether it is a gametophyte or not. Now what is gametophyte? You know, any structure that is capable of forming a gametes, this is what we call gametophyte. Now let's discuss about the male gametophyte or the very status of the pollen grain. You know very well, the pollen grain is going to develop from the microspore, right? Suppose if the question is asked that which is the first male gametophyte, the answer is microspore, right? So once the microspore gets converted into the pollen grain, so we can say it is a partially developed male gametophyte, isn't it? So the correct answer for question number 7 is option C, that is partially developed male gametophyte. Why we are saying partially developed? Because the moment we proceed from the microspore to the pollen grains, right? In the course of the development, what happens? Process of the development of the male gametophyte has already begun. So from microspore to pollen grains, mitotic division has already occurred. So that means the process has already begun. That's why we are calling them the partially developed male gametophyte, right? Okay. Let's talk about question number 7. Pollen grains are released from the anther due to option A, microbial degradation of epidermis, option B, activity of pectic substances of endothecium, option C, increase in the number of middle layer of anther wall, option D, activity of tapetal cells of anther wall. Again, it is a classical questions. So once again, I would like to say that these questions has been figured number of times in competitive examinations in one or other form. Okay. Question is simple. It is asking about how pollen grains are released. Okay, and what could be the methodology for the release of pollen grains from the anther. We have in the previous questions, in one of the previous questions we have discussed, is enclosed by a covering or wall that consists of 4 layers, right? Epidermis, followed by endothecium, followed by middle layer, followed by tapetum. This we have already discussed. Now you know, all these layers have very, very specific functions. Okay, and since they have a specific functions, so accordingly their chemical nature is as per their functions. So if you see the endothecium, in case of endothecium, what we see that they are chemically made up of pectic substances, right? And since they are made up of pectic substances, so whenever the pollen grains meant to be released, then the crack develop in the pectic substances and eventually it helps in release of the pollen grains. Okay, so the very role of endothecium in anther wall, that it helps in release of the pollen grain from them. Okay, so for question number 9, the correct answer is option B, that is activity of pectic substances of the endothecium wall. Okay, have a look at question number 8. That which of the following statement is correct? You need to figure it out that in the given 4 statement, which is correct. Option A says, Ubisch bodies occurs in tapetum and it plays role in formation of pollen exine. Option B, Ubisch body occurs in middle layer and it helps and it plays role in the formation of pollen exine. Option C, Ubisch bodies occurs in endothecium and it plays role in formation of pollen intine. Option D, Ubisch body occurs in epidermis and it plays role in the formation of pollen intine. Now first of all, what is Ubisch body and where it is found? What is their role? So that is first we need to understand, then we can figure it out that what are the statement that is correct or incorrect. Now Ubisch body is a typical structure that is found in the tapetum of the anther wall, and I am sure you remember that tapetum was the innermost layer of the anther wall. So Ubisch bodies are found in the tapetum. Okay, and Ubisch body plays role in the formation of exine of the pollen grain. Okay, we know that the prime role of the tapetum is to provide nutrition to the developing microspore. Okay, but in course of development, what happens that it is the Ubisch body that helps in the formation of the exine, or in other words, actually chemically it helps in the formation of sporopollenin, because we know very well that the pollen exine is actually made up of sporopollenin. Okay, by the way, I'm sure you know that sporopollenin is the toughest material, right? So the pollen grains exine is actually chemically made up of that is sporopollenin. So the correct answer for the question number 10 is option A, that is Ubisch body occurs in tapetum, right, and it plays role in the formation of the pollen exine. Okay, so that's all for this session. In the next sessions, we will be discussing couple of more important questions of reproduction in plants, right? Thank you.